Welcome back to another exciting episode of Dr. Bio. So this is going to be a tutorial series on GATE Biotechnology, and we are going to begin with the first module, that is biochemistry. So in this particular video, we are going to learn about the concepts and types of biomolecules. We are going to learn about carbohydrates and their biochemistry. We are also going to learn about proteins, amino acids and their biochemistry. Dr. Bio is a unique channel in YouTube where you're going to get all this information, all these tutorials for free, and it's going to help you in your preparation for GATE Biotechnology, DBT-JRF and other kind of examinations. So let's begin our learning. So what is a biomolecule? A biomolecule is an organic molecule which is predominantly found in biological systems. As we speak of biological systems, we speak of cells, we speak of multicellular organisms such as the insects, the plants, the animals, and all these biomolecules constitute the very basic of our living. So biomolecules can be broadly classified as proteins, carbohydrates, lipids, and some high energy compounds, and also nucleic acids. So the basic of biomolecules is that when we have a single molecule, we call it the monomer. When two biomolecules are joined with a covalent bond, we call it a dimer. When we have three biomolecules of the same type or different types in combination, we have a trimer. And we can have 4 units as a tetramer, we can have 5 units as a pentamer, and we can have a long chain of biomolecules. It can be carbohydrates, it can be proteins. We have a general term for that. We call them polymers. So as I said, lipids, nucleic acids, carbohydrates and proteins, these are the primary biomolecules which are found in living systems, and now we are going to study them one by one. Before going into any of the biomolecules, something which is very essential for us to understand is water, the biochemistry of water. Water, as you know, is made up of 2 oxygen atoms and 1 hydrogen atoms covalently bonded in such a way that there is a 104.5 degree angle between the hydrogen atoms, which creates a sort of a dipole where the hydrogen atoms are partially positively charged and the oxygen atom is partially negatively charged. It's also known as delta positive and delta negative. Water is the most abundantly found biomolecule in our living systems in the planet. All the essential other biomolecules are soluble within water. So water is also known as a universal solvent. It is also an important component of biological reactions such as hydrolysis. Water stabilizes protein and nucleic acid structures because of this dipole moment which is created by the partially positive hydrogen atoms and the partial negative oxygen atom. It creates a dipole moment for which all other kind of weak stabilizing interactions such as hydrogen bonds can form. Hydrogen bonds are very important in biological systems because they stabilize DNA structure, they stabilize the dissolving of any organic molecule in water. So, and biological buffers are also very important. For example, the blood in living organisms is made of bicarbonate buffer. When we work in molecular biology or cell culture, we use different kind of buffers, biological buffers such as HEPA, we use biological buffers such as HIPIS, we use biological buffers such as the phosphate buffer. All of these buffers, the principal component is water. So water, it acts as a biomolecule, it acts to stabilize other biomolecules within the system. So it is very important that we start with the basic understanding of water. Let us take a look at this picture. Here we can see what are the types of biomolecules in our systems. So one, we have carbohydrates. Carbohydrates are made up of sugar units, which can be glucose, fructose, galactose, any kind of sugar units, which are linked together by glycosidic bonds. So when we have 4 or more sugar units linked together, we call it a polysaccharide. So one class of compounds are carbohydrates. A second class of compounds are lipids. Lipids are important because They are the components of all cell membranes. I forgot to mention carbohydrates are essential because they provide us energy. They are present in the food we eat. They are present in the DNA strands, the backbones of the DNA strands. So carbohydrates are essential. Lipids are also essential. There are several kinds of lipids. In lipids, what we have is esterification reaction of fatty acids and glycerol. When three fatty acid molecules are bound together by aldehyde molecules such as glyceraldehyde, we have something called a triglycerides. So triglycerides are the basic building block of fat molecules or lipids. Now, the next important thing that we have in biomolecules are proteins. Proteins make up almost more than 60 to 75% of all cells and all the organelles in our system. Proteins are composed of chains of peptides. Peptides are itself broken down into amino acids. So if we take at the basic look of proteins, we have the basic unit which is amino acids. Then we have nucleic acids. Nucleic acids are the blueprint of DNA and RNA, which are the genetic material found inside the living cells. DNA and RNA are composed of sugars and sugars linked with nitrogenous bases. So nucleic acids can be classified as a separate class of biomolecules where we have ribose or 5 carbon sugars as well as nitrogenous bases, purines and pyrimidines. There's also another class of compounds, high energy compounds, which are also known as ATP. ATP are the energy currencies of the cell where, for example, glucose is taken in by the cell and the end result is the formation of ATP. In ATP, we have an adenosine group linked with 3 inorganic phosphate groups. So whenever these energy demanding phosphate groups are broken down, it liberates a lot of exothermic energy. So high energy compounds include ATP. Till now, whatever we were discussing, it was a general idea of what are the different types of biomolecules in our living systems. Now we will have a closer look into one of the main classes of biomolecules, the carbohydrates. Carbohydrates have been so named because of the word hydrates of carbon. They generally have the chemical formula CH2O whole N, where N has to be greater than 3. The simplest carbohydrates are 3 carbon compounds like glyceraldehyde, and the most abundantly found monosaccharide molecules are the glucose and the fructose, these kind of molecules. So let us have a brief look on what carbohydrates are basically. So we can divide carbohydrates into 2 different classes. One is simple carbohydrates and the other is complex carbohydrates. Simple carbohydrates are the ones which are usually in our dietary system, like glucose, fructose, the sugars which we actually metabolize, and complex carbohydrates are part of systems of storage of carbohydrates like in muscles. We'll come to that later. In simple carbohydrates, you can have monosaccharides or simple sugar and disaccharides where 2 sugar units are linked together. Monosaccharides are glucose, fructose, galactose, and disaccharides are maltose, sucrose, and lactose. Complex carbohydrates include starch. Starch is one of the main components of foods like potato and rice. And starch is broken down in our stomach, and ultimately it results in disaccharides and monosaccharides being absorbed in our system. Another important storage carbohydrate is glycogen. Our bodies store energy in the form of glycogen in the muscles. And fibers, the various fibrous plants like coconut and jute, these fibers are also carbohydrate polymers. A simple classification of monosaccharides and disaccharides can be, as we can see in this picture of yellow and blue. On the top we have sugars based on the number of carbon atoms, like triose sugars contain 3 carbon atoms, pentose sugars contain 5 carbon atoms, and hexose sugars contain 6 carbon atoms. On the left hand side we have a classification of aldoses and ketoses. Aldoses are those carbohydrates which contain an aldehyde group, which is a CHO group, And ketoses are those carbohydrates which contain a ketone group or C double bond O group. So first we see aldoses, which is a triose sugar, that is glyceraldehyde, 3-carbon compound with an aldehyde group. We have pentose sugar ribose. Ribose is one of the main components of DNA. Ribose is a pentose sugar, it is an aldolase. And hexose sugars include glucose and galactose. Similarly, ketose sugars include dihydroxyacetone, which is the smallest, the smaller monosaccharides, and the 5-carbon version of the ketoses is ribulose, and the 6th version is the fructose. So we can have different classification of carbohydrates based on the number of carbon atoms and whether they contain an aldehyde group or a ketone group. Cyclization of monosaccharides. Now naturally, monosaccharide units which are longer than 5 or 6 carbon atoms do not remain in a chain in a solution. So when they're in a chain form in a solution, they automatically react. One of the aldehyde or ketone groups of the monosaccharide reacts with another carbon of the same monosaccharide sugar and forms a cyclic structure. This process is called cyclization of monosaccharides. For example, you can see glucose is a 6-membered monosaccharide. So whenever it is present in a solution, carbon number 1, which is an aldehyde group carbon, reacts with carbon number 5 of glucose and forms a 6-membered ring. So this is called cyclization of monosaccharides. Go a little bit more details into the chemistry of these. We have isomers. So what are isomers? Isomers are compounds which have the same molecular formula but different structures. So they can be widely differentiated into constitutional isomers and stereoisomers. So what are constitutional isomers? Constitutional isomers are isomers which differ in the attachment of atoms. For example, if we take 2 molecules, glyceraldehyde and dihydroxyacetone, both are 3-carbon compounds, but they differ in the position of the keto group. Stereoisomers are compounds where the atoms are connected in different order but in different spatial arrangement. They can be further divided into enantiomers and diastereomers. Enantiomers are compounds which are non-superimposable mirror images of each other. A classic example here is glyceraldehyde, L-glyceraldehyde and D-glyceraldehyde. In L-glyceraldehyde you can see the OH group faces the right. In D-glyceraldehyde you can see the OH group faces the right, and in L-glyceraldehyde the OH group faces the left. So they are non-superimposable mirror images of each other. We also have diastereomers, which are isomers that are not mirror images. For example, maltose and glucose. We also have a further classification where diastereomers are also differentiated as epimers and anomers. What are epimers? Epimers are 2 molecules which differ in the position of several asymmetric carbon atoms. For example, in D-glucose and D-mannose, the molecular formula is the same, but the attachment of the H and OH groups, the spatial arrangement of the hydrogen and hydroxyl groups of different carbon atoms are different. Also anomers. Anomers are isomers that differ in asymmetric carbon formed only during the closure of ring structure. So these are the biochemistry definitions which can differentiate between the different kinds of carbohydrates. The aspect of carbohydrates is that they can form 2 types of rings. For example, if we take D-glucose, D-glucose exists as a pyranose sugar and a furanose sugar, maybe simultaneously or differently. So what happens is that when the skeletal structure of D-glucose converts into a 6-membered ring, we call it pyranose. And whenever it converts into a 5-membered ring, as it is found in the sugar of DNA and RNA, it is called a furanose form of sugar. So the monosaccharides can be broadly classified into 5-membered rings or the furanose rings, and the pyranose rings or 6-membered rings. 2 units of monosaccharide sugar are linked together using a special type of a covalent bond which is known as glycosidic bond. What happens in a glycosidic bond is that one hydroxyl group of one particular sugar reacts with another hydroxyl group of a different sugar, and one molecule of water is released in the process, and there is a bond formation which is a C single bond O single bond C bond. So this particular bond is called a glycosidic bond. And whenever suppose we are having dietary carbohydrates such as starch reaches our stomach, and then finally the enzymes of the stomach digest starch into 10 units, and finally into disaccharide, and finally into monosaccharides. So what happens during these lytic processes? What happens is this glycosidic bond is attacked, and a molecule of water comes into the picture, and the water gets divided into H plus ions and OH minus ions. H plus ions reacts with one of the hydroxyl groups, and the other OH minus ion reacts with the other group, forming two molecules of monosaccharide. So this process is called the breakage or cleavage of a glycosidic bond. Glycosidic bonds can be of two types, alpha glycosidic bond and beta glycosidic bond. When two same sugar units are combining, such as one molecule of glucose and another molecule of glucose. So a bond between two glucose molecules is an alpha glycosidic bond. A bond between two different kind of sugar molecules or monosaccharide molecules, for example, a bond between galactose and glucose, is a beta glycosidic bond. Attention to another class of biomolecules, proteins. Proteins are essentially the most important group of biomolecules and the most abundant group of biomolecules which exist in the living systems. Everything, right, the food we eat, the enzymes in our stomach which digests the food, the cornea of our eyes which make us see things, everything is protein. So proteins are the amino acids are the building blocks of proteins. So first we have to study what are amino acids. Amino acids are one kind of acids which contain NH3CH C double bond O group, where the alpha carbon is the carbon from where the R group emerges, and we have an alpha amino group attached to the same alpha carbon, and we have a carboxyl group. So in brief, an amino acid is a molecule which has a carboxylic group, an amino group, and an R group attached to the same carbon, which is known as the alpha carbon. The R groups can vary, and it can be a simple hydrogen, it can be a more complex group. Accordingly, the amino acid names will change. So we'll come to that in our next slide. Amino acids are bound together using a peptide bond. A peptide bond is a very critical topic in biochemistry because protein structure depends on the spatial arrangement of the peptide bond. So what happens in a peptide bond? You know, the alpha carbon contains a C double bond OH or a carboxylic group. Also, another amino acid, it contains carboxylic group as well as an amino group. So when the hydrogen of the amino group and the hydroxyl group of the carboxylic group combines and releases a water, there is a C double bond O NH bond forms. This is known as glycosidic bond. The amino acids are various in their shapes and sizes and in their properties, but one thing which is common is that whenever they are in a protein, they are always held together by this peptide bond. By definition, we know that amino acids are small organic molecules which have an alpha carbon linked to an R group. The same alpha carbon is linked to a carboxylic group, and the same alpha carbon is also linked to an amino group. Such molecules are known as amino acids. Now, amino acids have two kinds of classifications. One kind of classification is based on the properties of the R group. So R group can be different types, and accordingly the property of the amino acids will change. There are accordingly around 20 naturally occurring amino acids in nature, and all of them are represented by a 3-letter code and a 1-letter code. So both are acceptable. So let's take a look at what are the different kinds of R groups. The first group is non-polar R groups. Non-polar word means that it is hydrophobic. It does not like water. It does not mix well with water, and so when there is a solution of protein containing a lot of hydrophobic amino acids, they tend to be in the center of the protein, leaving the waters not in contact with them. So what are the residues which are hydrophobic or non-polar in nature: glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, and phenylalanine. So tryptophan and phenylalanine, these two are special because they have an aromatic R chain. That means their side chains are large and contain aromatic rings. So what are the polar amino acids? What are the polar R groups? Polar means that they are water loving, they are hydrophilic, and whenever they are in solution, they tend to be close to water. So what are the different R groups which have this polar behavior? So we have serine, represented by SER or simply S. We have threonine, tyrosine, asparagine, and glutamine. Tyrosine is also an aromatic amino acid. Then we have charged groups. Charged R groups are those where the overall charge on an R group is either positive or negative. So positively charged R groups are lysine, arginine, and histidine. So these molecules have a tending overall positive charge. And there are negatively charged amino acids, aspartic acid and glutamate. Also another classification of amino acids, but this one is based on the fact whichever amino acids are required by the body and the body can produce it, or the body cannot produce it and it has to be taken through diet. So based on this concept, amino acids are classified as essential. So what are essential amino acids? Essential amino acids are those which are not synthesized by the body and has to be taken through diet. These include histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Then we have conditionally non-essential amino acids, which means that essentially the body can produce them, but in certain conditions they cannot and has to be taken through diet. These include arginine, cysteine, glutamine, glycine, proline, and tyrosine. There are certain amino acids which the body is capable of producing itself. These are alanine, asparagine, aspartate, glutamate, and serine. And basic properties of amino acids. Amino acids are a very interesting class of molecules in the sense that they can exist as a positively charged group or a negatively charged group during different pHs. So an amino acid contains two ionizable groups. The amino group, from there a hydrogen can be released, and a carboxyl group. From there also another hydrogen can be released. So if we take a journey of an amino acid from a very acidic environment to a very basic environment in a way that the pH is gradually increasing, we will find that at a point will come where the carboxylic group will release a proton, and initially the molecule was positively charged at low pH, and then when a hydrogen atom is released at pH around 2 to 2.5, it contains a positively charged and a negatively charged group. So now this molecule is called a Zwitter ion. A Zwitter ion is a molecule which contains a positively charged group and a negatively charged group at the same time. Here the amino group is positively charged and the carboxylic group is negatively charged. So the net charge on the molecule is 0. When we further increase the pH and it goes to a basic point, another hydrogen atom is loose. This time the amino group loses the hydrogen atom. So now the molecule becomes overall negatively charged. So it becomes from acidic to basic, and the charge increases from plus 1 to minus 1. So every amino acid basically has got two ionizable groups. Acidic and basic properties of amino acids are dictated by a wonderful equation which is known as the Henderson-Hasselbalch equation. Henderson-Hasselbalch equation states that the pH is equal to pKa plus log concentration of acid divided by concentration of base. So this particular equation is very important in the sense that a lot of numerical questions are asked and graphs are given in various examinations. Suppose to calculate the concentration of the acid if the pH and pKa is given, or to calculate the pH if the pKa and the concentrations of the acid and bases are given. So for numerical questions, the Henderson-Hasselbalch equation is very important. Titration curve of glycine. So glycine, as we know, is the smallest amino acid. It is the simplest amino acid. So it is often used as an example to study the shift of change of positive to negative charge of an amino acid when the pH increases. So we are going to understand what buffering capacity, buffering regions are. So suppose we take an example where we have a bucket of water and we dissolve glycine molecules in that water, and then we are adding small amounts of alkali like potassium hydroxide or sodium hydroxide to the solution, so that the solution gradually becomes from acidic to alkaline. So what will happen? So initially, in a region where pH is around 2.34, it's a basic pH, it's an acidic pH region. Whenever we increase the concentration of alkali, there will not be a dominant shift in pH. So this region I indicated in blue is one buffering region, denoted by a value called pKa. So pKa is a measure of how effectively the carboxylic group of the amino acid is releasing the hydrogen atom. So at this point, it has got some buffering properties, and if we add more base, the pH of the system will not change. After that, there will be a gradual increase in pH until it reaches a basic pH, and still if we go on adding alkali, another protonation will occur. This time the amino group is going to release another hydrogen, and the overall charge on the molecule is going to be negative. So at this region around pH 9.6, there is also going to be some buffering. So there is a dual buffering process for amino acids. And the pi of the protein, which is shown in the middle as 5.97, is a point where the amino acid is neutral in charge. So this pi value is obtained by pKa plus pK1 plus pK2 divided by 2. So pi is a zone or a pH where the amino acid is neutral. We come to the end of another exciting video on Dr. Bio, where you have learned what are the basic biomolecules, what are the different types of biomolecules, carbohydrates, lipids, amino acids. There's going to be more nucleic acids and cofactors which we are going to cover in our subsequent videos. So please subscribe and put your comments, do write to us, and we'll be happy to talk to you, and happy learning.